Okay, so yes, most of the people have already joined, and I'm going to start then. So, well, first of all, hello everyone. My name is Yazu, and I am co-hosting this event with my friend Paul. We are happy to have you joining our kickoff event for our event series, iGEM Phototrophs Overgrow the World. I guess I should apologize first for the some technical problems that we had regarding the timetable. It seems that some people received wrong information on the starting time. We got a bit confused with the time zones, and we hope it didn't cause too much trouble. And well, we are planning a series of meetups during the iGEM season to help teams getting started with phototrophic organisms and to provide useful information, useful tips throughout the year. Also, we want to connect the iGEMers and create a community for phototroph teams inside iGEM. Now I would like to introduce the two organizing teams. On the one hand, there's the Bielefeld-Sebitech team.
So our team has no background of working with phototrophic organisms, and our PI and supervisor are very passionate about all kinds of bacteria, but they have never worked with plants or something that makes photosynthesis. And also, all previous teams from Bielefeld never worked with phototrophs. So for our team, the collaboration is already quite helpful. And on the other hand, we have the Marburg team.
Well, that would be me. While previous teams from Marburg have already worked with Cyanobacteria, for example, and we are very lucky to have PIs who are experienced with phototrophic organisms. It still has been very challenging to work with the plants, and we hope to learn a lot from you guys in the coming months. So moving on. So far, there have been very few iGEM teams that worked with phototrophs. And this year, for example, there are around 363 teams in total, and only 32 are working with phototrophs. With this collaboration project, we want to improve the work with phototrophs for current and future iGEM teams. And well, because there are so few phototroph teams every year, we believe there is a lack of substantial resources to assist with their specific needs. And this small number of teams is, we think it's not proportional to the huge potential of phototrophic organisms. To incentivize this field, we aim to bridge the gap and address this lack of resources in the community for teams who want to start out.
Yeah, and the aim of this collaboration is to get all of our teams to work together, and participating in this and the upcoming events, you can fulfill the silver medal criteria for collaboration, for example. Also, we will provide you with a fancy sticker for your wiki that you, so a sticker that you participated in these events. And today's event is according to the motto, Getting Started. And to do so, we invited several guest speakers. At first, we start with alumni from a successful phototroph team. Then we have our two main guests, Professor Ilka Axmann from the Heinrich Heine University in Düsseldorf, here in Germany, and Professor Jennifer Brophy from Stanford University. And after a short break, we have the part where we dive into methods of transformation and cultivation. This is split up for cyanobacteria, algae, and plants. And the breakout sessions then are followed by our elevator pitches. Then we included a troubleshooting session. And after this troubleshooting session, we will move over to the fun part, the game night. We already have a rough plan for the 3 upcoming meetings that will come after this meeting. And so there will be 3 more meetings every 3 to 4 weeks until the beginning of September. The topics will most likely be cloning and part standardization, measurement and standardization of cultivation, and the big question, how to make the world a better place. We think that especially the last two topics offer great opportunities for all teams to participate deeper than today, and thus to contribute to our iGEM Phototrophs community. If you have an idea how you can contribute to this project, please contact us. And this contribution in our project could, for example, be something like a workshop that you make on a specific topic. This collaboration in general has three main parts. The meetup series, where we learn more from specialists and from each other. The Slack workspace to keep in touch with each other and share information, and a Phototrophs handbook. This Phototrophs handbook that will contain the knowledge we gather here, and it should serve as a starting point for future teams following the same steps as we do. The table of contents you see here is just a very rough preliminary draft of our ideas, what we want to include. At this point, I want to come back to the meta criterion collaboration. I mentioned a minute ago that you can fulfill the meta criteria and get the sticker for the wiki if you contribute to the meetup series. One other way to fulfill the criteria is to help us with this handbook. If there is a topic where you collected a lot of data and gained experience, you can write one of these chapters if you want. So the iGEM Phototrophs community is not only about listening to interesting presentations, but also helping each other out, sharing information, and not only for this iGEM season, but also for future iGEM teams who will then not have the same struggle with their projects as we have. And I think that by now I have talked enough, and we can move on to the first point in our schedule, the presentation by the iGEM alumni from the team Kaiserslautern 2019. And they will be introduced by René.
So, hello everyone. Also hello and welcome from my side. I'm René. I'm the instructor of the iGEM Marburg team, and I'm also like a PhD student in Marburg at the at the Max Planck Institute, also working with phototrophic organisms. And I'm very happy that these two teams are organizing this event series together, because I think it's very valuable for the iGEM community. And with that, I would like to introduce to you Doro and Adrian from the 2019 Kaiserslautern team. They have been studying in Kaiserslautern for their bachelor's and also continuing there. And yeah, they have participated in 2019 as the first team of their university. And actually, I met them already in 2018 when they informed themselves for like how to participate in iGEM, and there I already got in contact with them, and later on I was also the mentor of their team, so I know their project and their team quite well, and I'm really excited that we got them for today, and they will present you like what comes with the fact, or like how to run a Photoshop team, and like what their project has been. I don't want to tease it a lot, and therefore I would say the stage is yours.
Thank you. So, I hope you can now all see the presentation, and again, thank you for the nice introduction, René. Today, we would like to show you what the most important points of a successful plant project are based on our project and what we have learned in 2019. We participated as 9 students in the undergrad category in iGEM under the title Klami Yummy. And as already mentioned, we were the first iGEM team of our university and still managed to create a successful project, which in the end brought us the second runner-up in the undergrad category, as well as extra prizes for the best plant project and environment project. Our post and wiki also received awards. But first, let's talk about our vision. plastic continues to accumulate in our environment, posing an unknown threat to the world ecosystems. Traditional recycling of plastic is energy intensive, environmentally damaging, and results in a loss of quality of the plastic. In addition, plastic that has been exposed to the environment for a long time can no longer be recycled. We want to solve this problem by enzymatically breaking down plastic into its monomers and purifying them. These monomers are also the starting material for the resynthesis of PET and some other bioplastics. This could lead to PET actually being recycled permanently instead of the current situation where it is largely made from petroleum and ends up in the rubbish or worse in the environment after 1 or 2 uses only. This whole project should happen in Chlamydomonas reinhardti, secreting 2 PET degrading enzymes while growing with the plastic weight in a photobioreactor, as you can see in the picture. So here on the next slide, you can see the detailed degradation scheme for PET when it is degraded by the 2 enzymes PETase and MHETase. PETase breaks the PET polymers into the monomer BHET or directly into MHET, and the MHETase then can break it into the real starting material which are also used to resynthesize PET. These both enzymes are derived from cutinases and differ from other PET degrading enzymes in their high specificity for PET and mild reaction conditions, which allow the production of the proteins at the same time where the PET is degraded. These enzymes come originally from the bacterium Ideonella zacchariensis, which has been identified as a key player in a group of microorganisms that grew on plastic. And yeah, after the degradation of PET, only ethylene glycol and terephthalate remains as an end product, which then can be reused. So, but now we want to come to the actual topic of today. Why did we choose the green algae Chlamydomonas reinhardti as our chassis? Chlamydomonas is a well-studied model organism. As a result, many mechanisms in the organism are understood and many methods are established. This makes it easy to use it as a chassis for SynBio. Its ability to grow photoautotrophic makes it a theoretically very cost-efficient and environmentally friendly chassis. This is a characteristic that unites all phototrophic chassis and makes biotechnology as a whole more adapted to the needs of society, especially in the future. For a plant, the generation time of 8 hours is pretty short and allows us to test and realize a project more quickly, and so it can be used as a chassis in iGEM. Also, after project realization, a high growth rate promises a higher product yield. And at the same time, Chlammy can be handled in the lab like a bacterium. For example, when it comes to the cultivation in liquid medium, and the general experiments are not so complex when you are working with higher plants. Also, Chlamydomonas can grow in a bioreactor, which lowers the cost of implementation and makes upscaling feasible. In addition, there's the possibility for post-translational modifications, which allows the expression of more complex genes. Glycosylation in particular can be beneficial for plants. Also, it is different from the glycosylation in animals, it often seems to have a stabilizing effect on the lifetime of proteins, which was especially important in our project because of the short lifetime of the PETAs. Also, Chlamydomonas was a particular good fit for our project because it could be shown that it is not negatively affected by microplastic, but rather attaches itself to them. For us, this means our enzyme are secreted directly in the proximity of their substrate and can thus work particularly effectively. In addition, we also tested the degradation products of PET for their toxicity to Chlamydomonas and were able to show that they also do not negatively affect the growth of Chlamydomonas. What makes the cultivation of Chlamydomonas in the presence of PET possible? The main disadvantage of phototrophic organisms is in general their slow growth rate compared to bacteria and the time problems this causes in the iGEM competition. Therefore, it is extremely important that other parts in the project progresses quickly, and the creation of genetic part is the most effective way to speed up a iGEM project. All modern cloning methods have advantages over biobricks. And a common feature is that the combination of several parts in a single reaction which speed up the whole process enormously. In our case, we used the modular cloning system. This is already implemented at our university for Chlamydomonas, and therefore we could use the system easily. The modularity and standardization of the parts used in the systems makes the system flexible, and individual parts can be exchanged quickly, allowing to optimize our project while we getting our first results. And yes. General in our project, it took 4 weeks to get from the finished parts to the first analysis of the mutants because of the slower growth rate of Chlamydomonas. And with the Moclo system, we were able to produce new parts quickly and efficient, and therefore the cloning of new parts was never a limiting step for us. And also we had to wait these 4 weeks till we could start testing with our new constructs. We had enough to do in the meantime because we were still working on parts we had produced previously. In our project, we produced 78 parts and combined them into 35 different composite parts, which you can see in the background. And we can really recommend you to work with a modern cloning method, or general with modern methods that are highly efficient, and this can enable you to do complex iGEM project with phototrophic organisms, which would not be possible if these methods were not there.
So another important point that helped us enormously with our project was to structure our project. I know it sounds trivial, but especially when working with a phototrophic organism, it is very difficult to correct mistakes. And therefore, it is useful to divide your project into smaller steps. And the result of these investigations help you to quickly discover problems and to solve them and to adjust the project accordingly. And as I said before, it is important in general, but especially for your project, it can make a huge difference to go step by step. And to give you a short insight into our project, our main goal was to secrete the proteins of interest. And but we started with way smaller steps, with way smaller goals. And first of all, we tested whether the genes of interest could be expressed at all. And to do this, we took a cytosolic approach. And so we at first looked if this is possible, because we thought if everything else fails, we could simply lyse our cells to release the enzymes instead of secreting them. And realizing that the expression worked, we tested new constructs that contained secretion signals. And here it turned out that there were problems with the secretion, and more detailed investigations then showed that the enzymes got stuck, as you can see here, inside of the cells during the secretion. And in the meantime, we had thought about such a problem, and we worked on the optimization of our parts. And we used more secretion signals, so different secretion signals, and secretion enhancing SP20 tech. And indeed, this enabled us to successfully secrete our proteins of interest, as you can see on this Western blot. Another advantage of our project was the parallel approach, which means we simultaneously implemented our project in E. coli to demonstrate the advantages of our chassis in a direct comparison. For example, when it comes to glycosylation, that these have a positive influence on the stability of our enzymes. And at the same time, we used it to set up a robust analytical protocol for testing our enzymes before the Chlamydia, or the Chlamydomonas enzymes, could be tested. But one of the most important points why we did this parallel approach is that this E. coli project was kind of a backup for us, because if nothing had worked in Chlamydomonas, we could have focused on the E. coli results, of course. And honestly, as our first university's iGEM team, we were a bit overwhelmed at first, because we didn't know how iGEM works, and but we were able to overcome this with trying to work very closely with our university's phototrophic research groups. We also joined forces with other iGEM teams. So, for example, with the iGEM team of Berlin and the Sorbonne University iGEM team, because they have the same organism, they use the same organism. And we also collaborated with the iGEM team from Marburg to optimize our cloning process. And in this way, we exchanged knowledge kind of and helped each other. And and however, when you plan a project, well, when we planned our project, we also tried to understand why previous PTA's projects did not work or were not that successful. And so we kind of integrated knowledge of previous iGEM teams. And in terms of sponsoring, for example, the Munich team of 2018, as René kind of said, it helped us a lot. And without our mentor, speaking of René, we would never have made it to this competition as the first team. And all this and the help of experts made it possible for us to do a good project. And therefore, it is our advice for you, to don't hesitate to ask experts for support. And I'm almost at the end of the talk, but what I like to emphasize is that you can be proud of your work. you can be proud of your work. It is not easy to work with a phototrophic organism, but yeah, you are rising to the challenge, you know, because you know exactly the advantages of the organisms, otherwise you wouldn't use them. Yeah, and remember that with using such organisms, you can also have a positive impact and a lasting impact on society. And that's exactly what iGEM sees, because that's why there's a special award for best plant synthetic biology. And what we think also is also important, that you should encourage other iGEM teams or the iGEM community to use phototrophic organisms as well. To make it easier, for example, for future iGEM teams to work with our model organisms, so with Chlamydomonas, we have written a how to handle chlammy guide in collaboration with the Sorbonne University team. And yeah, with that, I would like to thank you for your attention. And if you have any questions, we would be happy to answer them. But finally, I would like to thank our sponsors and advisors, and especially our PIs and our instructors and our mentor, and because without them, the entire project would not have been possible. And if you are interested in our project and want to learn more about it, or you want to contact us somehow, here are a few links. Thank you very much.
Thank you for that great presentation. And yeah, I think we learned a lot. And if anyone has a question now, you can either raise your hand as a participant in the participant section, or write a Q into the chat. Then I would call you, and you can ask the question either in the chat or by using your microphone.
Can I just go out and say that that illustration of the clammy for the guide is probably the cutest illustration of an unicellular organism I have ever seen.
Yeah.
Thank you.
Do you have another question, or was this your only question?
No, actually, I have a question, which is, well, first of all, I want to say this really awesome, the presentation, your project also really, really interesting. And what do you wish you had done differently now with the benefit of hindsight? Looking back in your project, what would you change?
I think the part where we were not so good was the activity assays. So we could, in the end, show, yes, we can produce enzymes in Chlamydomonas that can degrade PET, and but it's really hard to detect this reaction because it doesn't follow Michaelis-Menten kinetics, and also there is not something like a color change or something you could track while the reaction is ongoing. And so we analyzed hundreds of HPLC samples and tried therefore to prove that our enzymes are really active. And a problem for us was that we didn't start this as structured as the work with Chlamydomonas, and therefore we had many experiments started, but we couldn't really get a good result. And this is the part where I would again say try to work more in steps and show that you have a small success and build up on that. And then, for example, though maybe you can go back to the PET degrading slide. BHET is also a substrate of the PETAs, and so you can use this to monitor the reaction. And we use this a lot, but with that, because this is not a monomer, an polymer, but only this one molecule, most of the different mutation we looked into didn't have an effect on the kinetics, and we didn't think about this before. And I think a better planning of a project is always really helpful and can save you a lot of time. And yeah, so I think this is a problem in our project.
Yeah, thank you very much.
Okay, there's one more question from Marvin.
Yeah, a very nice project. Did you were you able to get an idea on how long it would take to degrade a given amount of PET, for example, 1 gram or something like this?
Yes, we tried to calculate that, and it was not so good. So I don't have the exact number in mind, but we prepared something for our final presentation because we thought this will be a question we will definitively get. And I can say at this moment, it is not feasible to degrade PET with enzymes in general, and especially not under this mild conditions. And we used really in 2018, so 2 years after the enzymes were first published, there were not so many mutations that increase activity, but now they could already increase the activity over longer times. I think they could increase the activity 90-fold. So this is already a really great success. Most was going to the stability part, so that the enzymes are just longer active. But yeah, there's great progress, and we think we could implement easily new mutants in our system. And so this was also our answer, that right now it's not useful because it would be too, it's not cheap enough, but in the future, maybe something like this can work.
Oh,
Okay.
nice. Okay.
I don't see any further questions. So thank you again, Adrian and Doro. And I would say we will continue with our next speaker, which is Professor Ilka Axmann. And I will just briefly introduce her. She did her undergrad studies in biotechnology in Berlin at the TU. Then following a PhD also in Berlin at the Humboldt University. Then she did her postdoc at the Charité in Berlin. And since 2013, she went to Düsseldorf, was the junior group leader, and since a few years, she is now full professor for synthetic microbiology at the Heinrich Heine University in Düsseldorf. And with that, I hand over to Ilka.
Yeah, many thanks for your kind introduction, and of course, thanks a lot for the invitation. So I enjoyed the previous talk. So the plastics, to the algae who are eating plastic, I highly enjoyed these days. And yeah, it's amazing what can be possible in Phototrophs. So of course, I fully agree. It's a bit more challenging to engineer a phototroph than E. coli, but on the other hand, it's I think more acknowledged in the end. So that should be some motivation to go further. And I think that's the way to go. So the more people we are working on the phototrophs, I think the easier it comes that, yeah, we can learn them new tricks. Yeah, and I would love to do some advertisement for the cyanobacteria. today, because that's a major topic of my lab. So I'm head of the synthetic microbiology in Düsseldorf in Germany. And yeah, we are focusing on the cyanobacteria. And yeah, of course, I do not have to mention this maybe here, but as we all know, life on Earth is driven by the power of photosynthesis. So that's the only way to transform that chemicals into life. So to build up organic matter, which all of us rely on. So that's the basic system. Our Earth provides food and feed to all of us heterotrophs. And to give you an impression about the microalgae players in that game, so it is estimated that up to or even more than 50% of carbon cycling belongs to the phytoplankton. So we have 3/4 covered by oceans, and that's a big amount of biomass. And yeah, the estimates are roughly 50 or even more than 50% of carbon cycling and oxygen production by phytoplankton, where a large part is cyanobacteria. So here in this picture, you see the Silicococcus, but even higher number are given to Prochlorococcus, which inhabit marine environments and are big players in these global cycles. Yeah, and these cyanobacteria are not only found in the oceans, so they are everywhere. So that's something which I find always surprising. So we started funny projects at our campus. So I asked students to run over our campus and take samples from everywhere, scratch some green, brown, whatever slime at stones, like you see here with the red arrows, or from small ponds, or these black stripes, some funny little organismal groups everywhere. Take them to a lab and try to cultivate them. And after weeks and months of cultivating them on, in this case, BG11, with and without a nitrogen source, we indeed found cyanobacteria there. So there are in these communities everywhere distributed on our planet, even at our campus. And they were really funny and, to me, beautiful structures appearing. You see here single-cell organisms, even dividing. Here are some packed versions, and we even found more complex filamentous cyanobacteria. And here, for example, even heterocyst forming ones, which are able to fix nitrogen, which I think is a very special feature of the cyanobacteria, which departs them from plants, because plants need nitrogen. Atmospheric one they cannot fix, but at least several cyanobacteria species are able to fix atmospheric nitrogen and are then players in the nitrogen cycles as well. Yeah, what we are doing in lab with our cyanobacteria is, we would love to engineer them in a way that they not only produce the basic compounds, let's say sugar and oxygen, but also some interesting metabolites of our choice, so that we train them new pathways of our interest. And we started by engineering a biosynthesis of plant terpenes in cyanobacteria. So maybe let's start like this. So cyanobacteria naturally encode the MAP pathway, which is a non-mevalonate pathway, which is also found in the plant chloroplasts, because the cyanos are progenitor, or the chloroplasts are related to cyanobacteria, so they are the progenitors of the plant chloroplasts. So we have at least a few similar pathways. One is the MAP pathway, and naturally cyanobacteria can produce the squalene, which is already an interesting component, for example, used as an adjuvant in vaccines. So actually there's a big interest in squalene and that can be found, of course, in other organisms as well. For example, in sharks, but nobody likes to kill millions of sharks, so it would be cool to have these components. Naturally, you can derive it from the cyanobacteria, but if you put further interesting genes connecting to this natural pathway, for example, squalene epoxidase and cyclase, a specific one, from a plant, then you're able to derive at certain plant terpenes. So that was pretty easy doable in cyanobacteria. So we were really surprised. We managed to do it, I think, almost half a year. We were able to add simply 2 genes from Arabidopsis into our cyanobacterial system, which is Synchocystis species. And indeed, we found by mass spectrometric analysis, we found the oxidosqualene as an intermediate, but also we found almost all of the plant terpenes we targeted. So that was really encouraging for us, giving a kind of proof of concept that we can derive at these plant sterols, but even further unusual triterpene scaffolds, which are mainly found in plants. So they are the main source of all these components. However, yeah, we outcompeted our partners in Jülich a little bit. So they were using Rhodobacter. So our levels of triterpenes were a bit higher than Rhodobacter. However, still fairly low compared to yeast, which is also engineered to derive plant terpenes. So we could not compete with the yeast values these days. So we thought about how to engineer the pathway further to derive at higher yields. And if you look at the map, at the metabolic map of this pathway, you immediately see that, yeah, the trick is to enhance either the MAP pathway to derive at, for example, the FPP, farnesyl pyrophosphate as an intermediate, and then go further to the squalene and to the terpenes. However, in doing so, unfortunately, you also derive at yeah, a side pathway, or you enhance also the side pathway, which directs to carotenoids of the cyanobacteria. So it's tricky to have your heterologous product enhanced without interfering with the natural pigment synthesis of the cyanobacteria, which is yeah, a very sensitive step, because you need the carotenoids. So if you would, yeah, cut here the pathway, they would die, so they cannot live without carotenoids. On the other hand, if you enhance some steps before, then you parallel enhance this natural pigmentation. So here a good balance is needed, and we had to think about how to find a good balance for this pathway, because, for example, if you overexpress, for example, the CRTE gene, you have the effect to, yeah, higher up also the pigments. So we try to use engineering a trick which has been used before. So we, yeah, try to engineer the CRTE gene. But as I said, you cannot knock out the gene, because then you destroy the carotenoids or the pathway deriving at carotenoids and others. So we used, kindly provided by the Hudson Lab in Sweden, the CRISPR-I system to not knock out, but knock down the enzymes. So that was one nice trick, and we were nicely able to not only switch it down, but to gradually regulate down the gene expression by using our inducible promoter system. We published last year, Annabelle in my lab, she engineered a very nice set of inducible promoters for cyanobacteria, for example, the well-known TED-R system, which is then upstream or regulating the DAD-Cas9 system in cyanobacteria. And by that, you can tune up and down the amount of dead Cas9, which is then targeted with the corresponding RNA, CRISPR RNA to the promoter of the CRTE, I think the promoter site of the CRTE gene. So we were able to gradually downregulate in stepwise the CRTE gene and then check how low it can become that we still have surviving colonies. Of course, we then switch down not only carotenoids, but also our product. But here, the trick was that we then were able to introduce a heterologous gene homolog to the CRTE, which is not metabolizing the path to the carotenoids. So the CRTE is metabolizing 3 different reactions in the pathway. So these 2 steps to FPP, but also the step to geranyl pyrophosphate, which then leads to carotenoids. But if you instead use the ISPA gene, heterologous one, then you only have these 2 steps. So you enhance the flow towards your plant terpene, but not enhancing the flow to your natural carotenoids. And that was, I found impressively visible by looking at the colonies, so which are normally Silicocistis looking green, blue-green, as this Cyanobacterium, blue-green algae. But our engineered strain, which pumps most of the FPP into the heterologous product, the plant terpene, looks bluish. So we obviously downregulated carotenoids and arrived at this blue phenotype, and impressively also not only changed or lowered the carotenoid pigmentation, but we enhanced, I think, up to 5-fold the plant terpene accumulation. So it was pretty successful engineering, at least in our eyes. Of course, it wasn't done by myself. So I have a very brave team caring for these little cyanobacteria. Main of the work, as I said, was done by Annabelle, Max Dietsch, and supported also by a lot of students, by Pia and many others. Anna helped by modeling, and as I already said, we had support from Jülich, where we got the genes and valuable parts for the pathway. We derived the CRISPR-I system from the Hudson lab, which are really helpful and supportive when we ask for the parts and for the designing of the CRISPR RNA. And initial design was done by Dennis, who's now in Phototrophs, in the company. I will tell a few words next. And metabolic, so the mass spec analysis were done via our C+ mass spec platform, which is also very much needed if you do metabolic engineering. So I think we have now a good platform at hand with a good set of tools so that we can engineer the cyanobacteria to our needs, to our wishes, which is really nice to see. I've never thought to have this at hand in a few years. So it's really amazing that we arrived now at this stage, that we can almost do whatever we want with the cyanobacteria. So we have this great platform. Further, there are many, many different cyanospecies out there. So although you can engineer your host of interest, but on the other hand, as I showed in the beginning, there are so many cyanospecies around, so you could even look and run for a species which is maybe producing a very interesting component you're looking for. So it's also an option to use this natural large resource of cyanobacteria strains. And as I will show in the next few minutes, there's already large-scale biotech going on for cyanobacteria and microalgae. So that's a great resource, and several players, even globally, which make money out of cyanobacteria. So that's also possible. So here it's my list of examples. I think the classics is to have biomass from cyanobacteria, which are also called spirulina. So they are approved as food. So we are allowed to take them for food or feed, and it's mainly Arthrospira as a species which is used. You can use the pigments of the cyanobacteria as colorants, fine chemicals. Here mainly phycocyanin is a big player having or giving blue color, for example, to food. There are companies going into renewable energy, biofuels. One example is here ethanol production based on cyanobacteria. I will show you an example from our university where an interesting enzyme has been found, which can be used, for example, for cosmetics. And cyanobacteria are also known to produce a variety of toxins, mainly non-ribosomal peptides. And that is also a big emerging field for pharmaceutical industry. So 2 weeks ago, we had a very interesting, amazing talk showing that, for example, cancer treatment pharmacy is based on cyanobacterial toxins. So there's, I think, a further big potential into that. Yeah, just to give you an impression how that looks like, we would love to have a field or agriculture with the cyanobacteria. So here the DICE group is a big player, producing about 1,000 tons of spirulina every year, mainly in China and in the US, where they provide then the biomass in form of dried spirulina, or they also produce to a large extent the blue color. So the phycocyanin, which gave Smarties their blue look back years ago, where they were forbidden at the market. So the blue chemical color these days was not allowed anymore because of several health issues. And Smarties were allowed to come back using spirulina blue color. So if you look at your next package from your supermarket trip, have a look. Normally there is spirulina, or what's called, some plant-derived coloring is written there. Yeah, alginol is another example where people found, colleagues from Berlin found naturally producing cyanobacteria strain, naturally producing ethanol. And that went up to be bought by a US-American company called alginol, and they have a big production field, for example, in Florida, but also Mexico, where they have large-scale bioreactors running for microalgae production. And as I promised, I highly enjoy this example, making use of photolyase from cyanobacteria. So I thought, indeed, that could be one iGEM project. Unfortunately, it's already on the market, so you cannot use the idea anymore. But here, a professor from the Leibniz Institute, close to our campus, he found this enzyme, took it from the cyanobacteria, heterologically expressed it, encapsulated it in liposomes, and that helped against or repairing the UV mediated cell damage. And now you can go to your pharmacy and buy it. So it's on the market and should help against your red skin in summer. So maybe that is one, hopefully, very current example how you can make use of cyanobacteria and how the way it works. So as you know, your thymines on your DNA are treated or impacted by UV radiation and became thymine dimers. So it's a big problem, can also cause mutations of course, also skin cancer. But the cyanobacteria, as they are phototrophs and highly impacted by light and UV, they have a rescue mechanism. So they have these lyases, which then are able to metabolize the dimers into, again, monomers. And obviously that is working as well on your skin. So at least the colleagues showed that if you apply this lyase to your skin after sunburn, you can recover from sunburn again. Okay, if you like to learn further details on cyanobacteria, we have an online seminar series running, which is free. So please feel free to sign up and join us. They're also listed SynBio, so synthetic biology talks. I think Paula next on July will tell interesting stories for SynBio. There's also Alistair McCormick in September, for example, which I can recommend. And further interesting talks, so maybe Ralph for modeling is also highly recommended in terms of cyano research. And because previously your iGEM team Bielefeld told that they are not experienced with algae or cyanobacteria, I don't know if you realize that there is a meeting running in Bielefeld in September on algae biotechnology. So maybe that would be a chance to connect and network with the algae or microalgae community at your place. So this is a very impressive lineup. So I would say that these are the main players in algae biotech in Bielefeld. Soon, as I said, also for photonol, I see people, and highly recommended to reach out to this event. Julie will be there as well. Yeah, if you have further questions, ideas, or would love to come back to us, please send an email or follow us. You're free to have a look and further check our homepage. And I think Nick will do further advertising later on tonight for Cyano World. So there's also, I think, one initial idea to combine or to unite the community and to have a united power for cyanobacteria and phototrophic organisms. So it's something I highly appreciate. Yeah, and I'm looking forward to any questions.
Thank you for the talk. As before, like, you can write in the chat or raise your hand under the participant, and I would call you. I can also see first question, Yazoo. Or was this like just a...
Unfortunately, I don't see. So you have to, I don't know, scream or write in the chat. I might see something.
Okay, this was not a question, yeah. There.
Thank you. Very welcome.
Okay. Is there any question?
Question mark looks good.
Yep. Adrian?
I have a question, just if I got it right. So this sun milk or something like this, are they heterolog expressed? So
right, yeah.
Okay.
They are.
Wow,
it's just a gene derived from the cyanobacteria, but let me guess. I guess it's E. coli. They're using as a chassis for heterologous production.
I think this is really interesting that the people do not have a problem with that, because I think it's another nice example where we can see the difference between the food and stuff we put
Absolutely.
to our...
Exactly. As soon as there are no living GMOs in the play, it's not GMO anymore. So that's the good part of biotechnology. As soon as you purify to some extent, it's not GMO anymore, and you can eat it, you can put it on your skin, you can, I don't know, drink it. So there's no issue about it. So it's just if you have the cells And even microorganisms, I think, are not problematic. So plants are the big issue. So people don't like to have, I don't know, Frankenstein plants. So somehow that is scary for, at least for European market. But yeah, as you've seen, so you are not, how to say, you are not restricted to manipulating the whole plant. So you can take plant enzymes out as a gene or as a protein or as a pathway. So you are not restricted to work only in plants. So you can take pieces of plants or just their genes, and then it's still phototrophically based somehow, but it's not needed to work with the full plant. So that's a good trick. So people in Jülich, they're working on pathways from trees. So they extracted genes, pathways from a tree. And of course, you would never run the iGEM project on modifying or engineering a tree, I guess. But if you use the right tricks, you can also call it a tree project. So that's something you really have to think about. So what do you need and how to put it together that you will be ready in one summer. So
okay, there's
feasible.
There's some more questions. So first of all, maybe Michael.
Yes. First of all, thank you for the nice talk. And
yeah, welcome.
My question is, I hope I didn't miss it, but have you actually sequenced the isolated cyanobacteria during your course? I'm curious.
Yeah, good point. Yeah. That's a tricky part. So we had a hard time to isolate DNA from everything we found outside. So that's not that trivial, but we succeeded successfully, and we now starting to do 16S in the beginning. But on the long run, we're also planning to do full genome sequencing. Of course, it's a matter of financiation, but that's intended for a long run. And we are also cooperating with the strain collection in Braunschweig, with the DSMZ, and they are doing it professionally. So that's something we are trying to optimize, so that of course we can train the students, we can learn how to do, but on the other hand, also have feedback and support from the experts from the strain collection. But we found some preliminary results were really amazing. So we found some cyano species which were annotated as Brazilian type. So we have Brazilian species at our campus and such things. So that's quite encouraging to hunt further.
It's pretty interesting.
Yeah.
I'm also like now thinking about, isn't it actually possible that there are species that can be found in like many different countries?
I'm absolutely sure. So there are amazing publications from for example, New York Tube or some things, or I think London Tube as well, where they've found bugs from all over the world. So that's the way you can imagine that they are by touristic or traveling. They are exchanged and distributed, but I'm pretty sure, especially in terms of cyanobacteria, they are widely distributed, and they are not. So a lot of species are not that niche adapted as we think. So I think they can survive many different conditions and then easily distribute around. And I think you will find them all over the world. So I know from Microcystis aeruginosa, which is a toxin producer, I think it's almost, in every second lake in the world, you can find them in China, but also in Germany.
Okay.
So it's not that specific as you, as a biologist, think of in terms of species, that you have the elephant over there in Africa and I don't know, kangaroo in Australia. So it's rather broadly distributed, many species. Maybe not all of them are, but many.
Okay, maybe we do two more questions. What about Jorge, which is raising his hand for quite some while?
I'm sorry. I didn't see.
Yes, thank you, Renee, and also Ilka for such an interesting seminar. And also, my question, I think, will answer a question from Tristan and Likith in the chat, because it's related with actually most of the groups working with cyanobacteria work with model organisms like Synecocystis PCC 6803 or with the 7942 Synechococcus. So actually, till what extent do you think from your perspective that maybe moving forward to different chassis organisms are a great thing, not for maybe producing metabolites that are naturally produced, but actually to use them as a platform for creating maybe novel metabolic pathways or similar things? So what will be the extent in the coming years for this production platform, just solar foils, more or less? Thank you.
Yeah, good question. Many thanks. So that's also something I thought about. So to what extent we can, push the people to use further organisms. Of course, the limiting point is that you can genetically engineer them. So I was working with marine cyanobacteria, for example, Prochlorococcus, and we never, ever derived at a mutant. So then, of course, you are limited somehow, but also even there, I think it's just a matter of try and error. So you have to push it to the limit. So I think there are ways. So I remember my first supervisor who told me any bug can be engineered. So you just have to know how. And I think that's the point. So I think the more people are into it and are really interested in engineering specific strains or new strains, they will success. So I'm pretty sure. So there's a way. We are now pushing a bit more electroporation, which of course is not that easy at the beginning, but if you have first protocols at hand, I think these can be adapted to almost all strains. So I read publications, for example, for Atrospira, which of course would be nice to engineer. I think 1 or 2 labs succeeded, and that can be pushed. And we are also now trying to check if the strains we found or we get from somewhere, if we can engineer them. So to put some constructs inside. So I think that can be done. Of course, it becomes more elaborative if you go towards filamentous and even then group 5 cyanobacteria, which are too branching and really tricky to handle by itself. But you can engineer a plant, so why we should not engineer any cyanobacterium. So I think that's just a matter of time to enter.
Thank you.
I think that's a good point for the transition. Thank you again.
Welcome.
And now I would like to introduce our next speaker, where we actually switch gear a little bit from cyanobacteria to plants. And therefore, I would like to introduce Jennifer Brophy. And she studied or did her undergrad studies in Berkeley and continued her PhD at the MIT in the group of Chris Voigt, which many of you might know for his work. And now she continued with her postdoc in Stanford in the Dynany lab, where she's working on engineering plant roots. And with that, I would like to hand over to Jen.
I think everybody's back now. So we can continue with the original plan of the elevator pitches. Unfortunately, we didn't prepare an order because we were not sure which team will prepare one. So we didn't prepare an order. So maybe volunteers first.
We as the hosts should start. So either Bielefeld or Marburg.
I would be willing to start if that's fine for you.
Go for it.
Yeah. So obviously our first talk will be by Jonas from the iGEM Marburg team. And I think it's like 3 to 5 minutes and then up to 5 minutes Q&A.
Yes. I'm going to share my screen first. I hope I will be in time. You can also interrupt me if it's not.
Oh yeah, who is looking at the time?
I can do that.
Okay. So 5 minutes.
Yeah, maybe also, wait, do you already see my screen? Is it full screen for you?
Yes.
perfect.
Oh, you use the new photos. Nice.
Yes. Wait, I'm just looking for the pointer button. All right.
You're already finished with the video. Sorry.
We filmed it. All right. Yeah. So I'm Jonas. I'm one of the team leaders from iGEM Marburg, and I'm very happy to be able to present our project to you, which is called OpenPlast. So let's get started. So as you are probably all aware, climate change is like a really huge threat, not just to us, but also to all the crops that we really rely on. And it's been shown that it is already negatively affecting agriculture all around the globe. So I think probably in this space here, we can agree that engineered crops will have like a major role to play in the future of our agriculture. So we think that especially the chloroplasts of plants could be engineered, and they have a lot of great advantages to crop improvement in general. So why chloroplasts? You can engineer chloroplasts with really high precision because you do have efficient homologous recombination there, which is not really the case in the nuclear genome of plants. And also you can stack your transgenes in whole operons because of the prokaryotic origin of this organelle. Furthermore, there's like a pretty high level of gene expression in the chloroplast, partly also due to the absence of epigenetic transgene silencing. This was something Mark mentioned earlier, that in the nucleus you have that your transgenes are being silenced, but not really in the chloroplast. So I guess the most important factor is the improved biocontainment that chloroplasts can offer. So they are only inherited maternally, which means that all the DNA of the chloroplasts, so also possible transgenes, are very, very, very rarely passed on through pollen, which means that the risk of your transgenes escaping your field if you have your engineered crops is drastically reduced. So all of this is really nice, of course, but as you know, plant and especially chloroplast engineering is a very tedious process. So for chloroplast, for example, you don't really have these transient expression assays that were also mentioned earlier, at least not right now. So yeah, there are also very few tools available for the chloroplast in terms of genetic parts that you can use for engineering. Just to give you an idea, the whole iGEM registry has like 20,000 parts or even more, but only 18 of those have been designed to work in the chloroplast, and none of these 18 have actually been tested and characterized. So yeah, this is like a huge problem that we are going to tackle with our project OpenPlast. We are right now developing cell-free systems from chloroplasts of many different plants. Cell-free systems are basically in vitro tools that you can use as like a prototyping platform to rapidly characterize a lot of genetic parts. So they mainly consist of an extract that you can take out of an organism. In our case, it's not a whole organism, but the chloroplast. So what we do is we take the leaves of a plant, isolate the chloroplasts, lyse the chloroplasts, and then you have the extract with like the whole machinery that is capable of transcription and translation. So whole protein biosynthesis can be done in these systems. What we are going to do, apart from establishing these systems, is kind of using a machine learning guided approach to optimize the reaction mixture of these systems, because you do need to add a couple of other factors there. Furthermore, we will create a toolbox of Golden Gate-based standardized DNA parts that are designed to be used in the chloroplast of different plants and that will be characterized in our cell-free systems. So this toolbox will include regulatory elements from different plants, some of which are maybe a bit more exotic. So to give you an idea, we are working on chloroplast cell-free systems from tobacco, spinach, our favorite little green algae Chlamydomonas reinhardti, from maize, and also other crops like rice, soybean, wheat, and also one tree. So we are right now working to develop cell-free systems from chloroplasts of oak trees. So thanks to these systems being like whole in vitro tools, it is very easy to like scale up the number of reactions you do and really do multiplex high throughput experiments where you can characterize thousands of parts simultaneously. which is very, very hard to do if you want to do it in vivo in the actual plant chloroplast. So the idea is that by outsourcing this whole prototyping work that you have to do at the beginning of a SynBio project to the cell-free systems, you can really speed up the design, build, test, and learn cycle of plants in bio. And when you are done with your testing, move to the actual plant chloroplast. So we are OpenPlast, and we really want to help sprout a new generation of improved crops. Thank you very much for your attention, and I'm happy to take any questions.
Thank you very much, Jonas, for your presentation. I think your project is very promising.
Thank you.
Do we have any questions?
Well, I have a question. You mentioned something about ATP regeneration system. How do you want to accomplish this? Because it usually includes membranes and membrane transport. So how do you want to do this?
Yeah, there's a couple of different systems out there that can be used or are being used in cell-free systems. We will work with creatine phosphate and a creatine phosphokinase, which is kind of always putting like new phosphorus groups on your used ATP, so on ADP, to kind of regenerate that. There are some that are definitely better, to be honest, which are right now, I think, mainly used in other cell-free systems from E. coli and stuff, but they rely on some metabolic components that are not present in the chloroplast, so we sadly can't use those. Yeah, with creatine phosphate, you do have some problems in the long run, so these systems typically don't run as long as other systems with other ATP regeneration, but yeah, it does work.
There's one more question in the chat. What do you expect to see when isolating the chloroplast from different organisms? Would the machinery be similar?
This is a very, very good question, actually. So there are a lot of similarities when you look at the chloroplasts of different plants. Yeah, they are pretty similar. We are also testing something like this. So we do want to test certain genetic parts in different cell-free systems from different plant chloroplasts to kind of see whether there is like, or there are like universal parts that you could use in all of these. Yeah, so I think that there will definitely be differences, that maybe a tobacco promoter does not work as well in an oak chloroplast extract, but I could think that it does work, but maybe not as well. So yeah, it's a good question. So definitely maybe in the end we see that having one system is enough because it's pretty comparable, but we thought that it would also be nice to kind of show to people that you can do this with different plants if you want. So yeah, if you are working on a super exotic specific plant, you could still do a system for that, hopefully.
Okay, so next question from Marvin.
Yeah, you mentioned that you are trying to optimize the conditions, I think, with machine learning. How exactly do you do this?
Yeah, good question. I didn't really talk too long about that, but basically the idea is to do many different reactions. So it will be like maybe 3,000 something different reactions where you have a slightly different composition of all of these things, like for example ATP regeneration system, or how many amino acids do I add, all of these components, and then have the algorithm recognize, okay, if I change which parameter, how does this affect the expression in my system, and to kind of, yeah, let the algorithm pinpoint the perfect combination of chemicals. I think this is the rough idea. Maybe Cedric, if
yeah,
you...
I think I can also expand on this. So like, as you saw, like I mean, like there are like pretty much 20 different compounds in the reaction mixture, and as you might imagine, like changing the reaction mixture can have like a huge impact on the cell-free system. And like of course you can't test like every single combination. So for example, if we just use like 11 different compounds, varying those by just like 4 different concentrations, for example, you would get like a combinatorial space of like over 4 million different compounds.
That would be a lot.
Okay, so you try.
Just where did I stop?
What?
That you had 4 million possible.
Okay, 4 million different combinations. So yeah, I mean, this is not tractable, of course. Like even in a cell-free system, you can't test 4 million different compounds. So what you would do is like generate like a first, I don't know, like 384 plate of different compounds, and then use like an active learning approach. So you're just feeding like a neural network these data, and then it will test out like, okay, like how well does it match my expectation, and then it will suggest new data points. And like this whole method is based on a cell-free system optimization that was done in E. coli in 2020. And what they've shown like is that you can do it in just like 7 to 10 iterations. And on top of that, you can also use these data points. And if you have like a new batch, for example, or like a new organism, you can just redo the experiment by just measuring 25 to 30 different new concentrations which were suggested by the algorithm for like the highest learning efficiency, so-called. And then you can re-optimize even different batches, which is really helpful, I guess.
I think there's one more question, but we are well over time. The question is, would we like to continue or answer the question?
I can very quickly answer it while some other person starts sharing the screen. So basically, you have to store them at -80, and when you take them out, you will work on ice. But it works quite well to just refreeze them, rethaw them, use them again and again. And we want to test how stable they are if stored at -20, or if we freeze dry them and have them in a normal freezer. But we haven't tested that yet.
Okay, so thank you very much for this explanation. We can start with the next presentation.
Can you hear me now?
Yes.
Yep. Okay, so let's go. In our team, we have already prepared an elevator pitch that was too long, so we just make things short. We don't want to make no one bored right now, so I will start. So, first of all, what is actually our project about? Our project is an acronym for Cyanobacterial cyclic carbon capture, and this is the meaning of the 4C of our project. Actually, I'm not sure how many of us has already spoke with us, but if you try to reach our team, we are not as Madrid UCM team, we are as at 4C Fools in Instagram, Twitter, etc. So, what actually we trying to do for this year in iGEM? Of course, all of us are aware about the environmental problem we have. All of us know that atmospheric carbon dioxide levels are rising. All of us know that climate change is real, and maybe the way we have to produce things in our societies is not as sustainable as it could be, leading to a global unsustainability that we need to solve. And of course, synthetic biology has a lot of things to tell here, specifically the phototrophs. But with all of these problems, we think that phototrophs are a solution. Because right now we have solutions for them. For example, with the energy crisis, we have alternative technologies for energy production. We have wind turbines, we have solar panels, but most of these solutions has a problem. They are not for all sectors. You cannot, for example, power a plane or a truck with electricity only. You need fuels for that. And also, for example, they don't cover you can cover the energy part, but you cannot cover the matter, let's say. So if you want to produce fibers or pharmaceuticals or biofuels or plastics or whatever, you will need some technology that is not so pollutant and still being sustainable to just move from carbon-based sources, like carbon feedstock, for example, carbon dioxide, to useful products. And this is the point that I think most of the photosynthetic organisms iGEM teams are trying to build their own projects. And in our case, we are trying to do our own part. Our philosophy is trying to build a platform, like a technological system that allows for the production of chemicals, bulk chemicals for industry, like building blocks for making polymers and dissolvents, or almost any organic chemical thing usually produced out from petrol. So problem with this opportunity, and we think that actually there is an urgent need for a circular economy, and in this way we think that developing a stronger bioeconomy is crucial for a more sustainable future. And in this way we think that there is a very huge market for that. There is a lot of markets that are demanding actually more sustainable feedstock, are demanding more sustainable biofuels, and we think that taking inspiration from nature, just harnessing the potential of organisms like cyanobacteria, can actually help us to make a process that can work. So, okay, we can speak about everything, and I think that most of us still being familiar that right now we have a linear economy. We just extract we just extract petrol or petrol basically, petroleum, and we just convert it into fuels, chemicals, plastics, products, whatever, but during the process of conversion and in the end of this useful life is still becoming a waste material. Maybe it's carbon dioxide, or maybe just becoming some landfill there. So we want to close this cycle. I think that most phototroph teams tries to make this idea of circular economy. We're trying just to harness the carbon dioxide and become into useful products. And our technological platform has this idea. We just want to create a system that actually can convert carbon dioxide into any kind of useful product. And to do so, we think that the first step is not only speaking in general terms, but just focusing on details. We would like to produce biofuels, not because they are easy or actually they have a high selling price, but because we think that is a very, very good starting point that could be more catchy to people that know we're producing succinic acid or any other kind of strange building block. So this is the reason because we are selecting producing biofuels. Particularly, we are trying to produce butanol, a medium length chain alcohol, that can be directly used as a drop-in biofuel, just overpassing the limitations of ethanol or even biodiesel. It will be maybe a less pollutant and also a fuel that doesn't require you to replace all the automobiles that currently you can find in the streets, and has near 90% of the actually energetic density of gasoline, for example. So how are we going to do that actually? We have seen that a current panorama of microalgae or cyanobacterial or even plant industry is based on the same scheme. It's the same paradigm. You have to grow your biomass, you have to harvest your biomass, you have to extract the compounds of interest or process your biomass. For example, if you're growing cyanobacteria, you have to dry them out or lyophilize them. And of course, all of these small stages in the process cost a lot. Actually, growing the cyanobacteria or microalgae just ranges between 5 or 30%, depending on the system you're using, of the total cost of the production. But the most crucial step is harvesting and processing the biomass. So we thought that maybe there's a different way to do that, and this way is developing photobiocatalytic processes. It's just taking the organisms and making the organisms produce directly the thing we would like to obtain. So we are trying to do metabolic engineering to achieve this thing, because we think that if you overpass the harvesting and processing and you just need to purify your product from the growth media, you can improve the economic cost of the process, and actually you can start implementing these organisms within industry. To do so, we think that, of course, we just need an organism that can take sunlight, water, and carbon dioxide, that can hold harsh industrial conditions and can produce biofuels, chemicals, or almost any other thing you would like to do. To do this, we are just taking different proof concepts, and we are just sticking all of them together and see if they work actually. So to do this kind of project, we are mainly expressing a butanol biosynthetic pathway, and we are trying to redirect most of the carbon towards butanol production. But also we think that we don't need only a super producing organism, we also need a robust strain. This is the reason, because we have tried to reach this PCC 11801, a novel cyanobacteria discovered in India, that actually is not only that it grows really fast and it helps a lot during the lab procedures, because you don't need to wait for a whole week for the organism to grow, and also it's naturally transformable. But the point here is that it's also a robust cyanobacteria. It can help conditions between 30 to 42 degrees and can tolerate a lot of toxic metals and pollutants. So it's like the perfect organisms for industry, because it's not so sensitive as others. In addition, this organism, since it grows very fast, has demonstrated that their metabolisms can be easily engineered to produce compounds of interest at higher rates than typical model organisms. However, of course, this strain is covered by Nagoya protocol, and accessing to it maybe is not so easy as we thought at first sight. We have been in conversation with the National Biodiversity Authority of India. We have filled all the paper stuff, and right now we're stuck in the middle of a bureaucratic process, just waiting for their answer signs, probably the middle May or something like that. So let's see if we can take this strain. If not, we will just make things simple and just let everything in the theoretical part. And regarding the synthetic biology, very, very briefly, we're trying to do 4 things. We are introducing bypasses that will just redirect the carbon towards acetyl-CoA, that is the intermediate metabolite that we convert into butanol using a 6 enzymes pathway that is oxygen tolerant, is not oxygen sensitive as the Clostridium ones. We are also implementing a synthetic carbon fixation pathway that just takes the carbon dioxide from the media further, more than the Calvin-Benson-Basman cycle, and also it can incorporate the toxic metabolites like the glycolates produced during photorespiration. In addition, we're trying to spray chaperones, regulation factors, and a butanol selective transporter in order to improve the tolerance of cells towards the product, and also to secrete it efficiently to the outer media, incrementing the yields because the product will be less toxic to the cells. So at least this is what we're expecting to do. And basically, this is everything we can say. This is the roadmap we have proposed for our project. We are more or less right now here, somewhere in the middle of the way. We have started with the lab experiments a couple of weeks ago, and currently we are having some troubles with reaching enough funds for buying more materials, DNA synthesis, and so on. But in the end, we think that we will develop not only a metabolic engineering approach, but also we are thinking to develop tools that will be especially useful if we end working with the PCC 1101, because we will try to characterize new neutral sites. We are trying to develop a system for generative unmarked mutants without very, very long processes and without depending on CRISPR-Cas system. So in the end of the project, probably around September, you will have a lot of more news from us. And well, we also have analyzed the business perspective of these kind of projects, because I think that right now, yeah, it's the last slide, sorry. Okay. Basically, we have just seen that there is a very, very huge market, and maybe since most of the entrepreneurs would like to go to industry, it's important to say and to tell to industry people like, okay, there is market here. Maybe investing in this kind of technologies actually works. It is not just something of young people doing iGEM stuff. And this is all. Thank you very much for listening to us. This is our team, our sponsors, and if you have any questions, I'm here for replying to you.
That was long 9 minutes.
Yeah, sorry. I was actually checking the time, but I didn't have a watch, and I was like, maybe I'm out, and sorry for that.
Okay. Yes, there may be a short question as we are running out of time.
Yeah, maybe just quickly. So I was wondering if you don't get this strain from India. I also tried that in 2019. That was trouble. Are there other strains you would think about using?
Yes, actually, we thought about using the PCC 11901, I think, that is marine Cyanobacteria, will be weird, but has fungal contamination. And if not, we will use the standard PCC 7942, because Utex is not naturally transformable, and maybe we have some problems with that.
Yeah. Okay. Yeah. Then maybe on a regarding something Mark said in an earlier session, might be nice to already do that in parallel and maybe start in some of these strains.
Yes, actually, we would like to do that if we reach the nice one.
And maybe we can just continue that we don't get too much time, steal too much time.
So who's up next?
Just look in the chat.
Okay.
I think Isabella is first.
Yeah.
Oh, all right.
So you guys are seeing my screen?
Yes.
Okay. So first of all, thank you very much for doing all this. It's a pleasure to be here. My name is Isabella. I'm from Team iGEM Brazil. And today I'm here to present our projects for this year's competition, which is, Let It Be. This is the name of our project. So many of you might have heard of the sudden disappearance and death of bees. And this is a huge problem, as the lack of pollination negatively impacts the ecosystem, economics, and field distribution, since pollination is a huge ecological service, not only to forest plants, but also for crops. And researchers show that this problem is mainly caused due to the misuse of pesticide, especially of the class of the neonicotinoids. However, we can't just abolish its use, since they are very effective and important in agriculture. Therefore, our team is focusing on solving this problem by preserving both the lives of bees and preserving the good effects of the pesticides. So our idea is to create a GMO that is capable of degrading pesticides by the expression of a special enzyme. Not only that, but we will be using a pollen-specific promoter so we can preserve the life of bees by degrading the pesticide specifically in the pollen and eliminating its effects there while preserving its good effects in the remains of the plant. So we'll be focusing on imidacloprid, which is an insecticide from the class of the neonicotinoids. And for our construction, we are using a promoter for tomatoes pollen. And we are also adding a sequence for the enzyme CYP-CG1, which is a protein from the cytochrome family, which was found in a pesticide resistance gene in Drosophila melanogaster. But considering the COVID pandemic scenario in Brazil nowadays, we have a lack of access to laboratory, and this is an issue. So we are now delving into molecular modeling, which our goals are modeling the genetic construction to check CYP expression in plants. And we are also doing something we call beehive modeling, which we are gonna make a model of the environmental impact of our construction, simulating the contamination of hives before and after our project, which is Let It Be. So this is the overall idea of our project. I hope you all enjoyed it, and thank you very much for listening.
Thank you for staying so good in time.
No problem.
Is there any question? I already asked so many in the session before.
Yeah, we talked so much.
If not, we can also just continue. Okay.
Can I have a question?
Yeah.
What species or what type of bees are you working with? Or are you not really looking into the bees themselves?
Well, we were thinking about using mainly Apis mellifera, which are the common honeybees, because they are the most known and researched. But in tomatoes, there's a problem because there are specific types of bees. There are bombus. So we're also trying to investigate the behavior of these specific bees to the plant that we are using. But for the research of how much the contamination and toxicity works, we are using the common honeybees.
Thank you.
Nice. Yeah. And then we can continue, right?
Yes, can everyone hear me and see the slides?
Yes.
Good, cool. So hi, I'm Evelina, and I'm the co-head of laboratory actually from the iGEM Think Open team. And our project this year will be to desalinate seawater using cyanobacteria. 70% of the Earth's surface area is covered in water, but only 3% of it is fresh water, and 2.6 of it is inaccessible to humans. That leaves 7.8 billion people with access to only 0.4% of the water resources. However, there's a lot of seawater that can potentially be used in daily life with proper desalination. Current desalination methods are very energy consuming, not very cost effective, and there is also room for improving its efficiency. With all this in mind, we in the LEU iGEM team of 2021 want to use the naturally photosynthetic and halophilic cyanobacteria to desalinate water. We plan to use Cyanobacteria synecocystis PCC6803 in our project. Since cyanobacteria have a slow growth rate, we plan to include the halophilic and fast-growing bacterium Vibrio natriens as well. This allows us to test our DNA constructs for salt uptake in Vibrio natriens while allowing our cyanobacteria to grow. So how will we achieve this? I will just briefly explain the basis of our idea. In cyanobacteria, the export of sodium is ATP driven. So by turning the light off, we put the cyanobacteria under starving conditions, and by doing so, the export of sodium is inhibited. But at the same time as we stop the export of sodium, we also want to create a continued influx of sodium and also create an influx of chloride ions. To achieve this influx, we needed to use an ATP-independent driver. Our solution is to make the cyanobacteria express the protein halorhodopsin. Halorhodopsin is an ATP-independent chloride pump that will create an influx of chloride ions. However, early expression of halorhodopsin will slow down the growth of cyanobacteria. Therefore, we will use a promoter that's induced by darkness to control the halorhodopsin expression. So now that we have solved the issue of stopping the export of sodium ions and creating an influx of chloride ions, what is left is the influx of sodium. And this problem actually solves itself. Because due to the influx of chloride ions, a negative membrane potential is created, which will generate an influx of sodium. And through all of this, we will hopefully be able to successfully desalinate seawater using cyanobacteria. After the desalination has been achieved, we want to remove the bacteria from the water. Our plan to do this is by using the cellulose binding domain, or CBD in short, and expressing it on the surface of the cyanobacteria. Once CBD is expressed, we want to bind the bacteria to a cellulose matrix in order to separate the bacteria from the water. So these are the basic ideas for our project this year. Thank you for listening. And do you have any questions? I'll be happy to answer them.
Thank you very much for your presentation.
Is there any question on that project? So I would have a question actually. So how, or like, what are your thoughts on like maybe escaping bacteria which you cannot get out of the water after the whole procedure, or like yeah, I think it won't reach 100%, right? So yeah, what are your thoughts on like biocontainment and like on that kind of story?
So first off, we're gonna start on a really small scale, but the CBD planning is really in more of a planning phase and an initial idea. Mm-hmm. So we haven't really thought about what would happen. We just want to see, would it work, kind of, because we base a lot of our project idea on the Shanghai team of 2015. Which also dealt with cyanobacteria and desalination, but they didn't have any idea whatsoever on how to actually get the bacteria out. Mm-hmm. So makes sense. Yeah, so we just want to say, can it be done somewhat at least?
Okay, makes sense. Thank you.
Thank you.
I also have one sort question that I don't know if you have already thought about it, but have you tried to size the system? Like, for example, a system with this amount of cyanobacteria can process 100 liters of water in that time, or actually just further development after you just research on the synthetic biology part?
We actually have not yet been able to get into the lab. So we haven't done anything in the lab sense. We're getting our cyanobacteria on Monday, fortunately, because they were very hard to get a hold of. So for now, we're gonna do a calculation to see how many cyanobacteria is gonna be needed to desalinate like a liter of seawater. But so far, we still don't even have bacteria. So.
Cool. So just as a play of thought, what like directly came to my mind, it would be super nice if all of this works out and you have like in the end, it's like perfectly working, to have like, I don't know, some hardware on your roof where you just pump the seawater through when living to the sea, and then you just get fresh water out of it. That would be really cool, actually.
Yeah.
Like that idea.
we're aiming for that solution, but it's way long time away.
I think the next project is iGEM Toulouse, isn't it?
Maybe I've also a question.
Okay.
Can I ask it? So, do you expect problems with the cyanobacteria taking up salt and maybe dying from this?
Yes, we are a bit concerned with them taking up too much and dying. And we haven't really thought of a solution as of yet. But yes, it is a concern.
Okay, so I guess iGEM Toulouse can start.
Okay, thank you. So hi everyone, I am Maxence. I am the team leader of the iGEM Toulouse team, and I'm going to present you our project, which is named Elixio. So for this year's edition, we decided to focus on the issue of mute flowers. Mute flowers are flowers from which it is impossible to extract the fragrance, mainly due to the very low yield of the extraction processes for these plants. Nowadays, synthetic chemical molecules are used to try to mimic the smell of these flowers in cosmetic and perfume products. However, these molecules are not sustainable since they are petroleum-based and they are also produced in harsh physical-chemical conditions in very energy-consuming processes. These molecules cannot also be considered natural, which is a very big challenge in the cosmetic industry right now. There are many examples of these flowers, such as the lily, hyacinth, lilac, and the violet. For our project, we decided to focus on the violet, which is the symbol of our city, Toulouse. It is also flowers whose scent is very research for cosmetic and perfumes. So our project aims to produce an ingredient that would mimic the smell of the violet flower through synthetic biology. To do that, we focused on analysis that had the objective to see which were the main violet flower volatile compounds that are responsible for its smell. So while doing this, we saw that there are mainly two families of molecules. There are terpenes and also aldehydes. And therefore, our idea is to develop a synthetic microbial consortium between the cyanobacteria and the yeast, and to produce the two families of compounds that I just presented while using the physiologic characteristic of the two organisms. So the terpenes would be produced by the yeast and the aldehydes by the cyanobacteria. This system also allows us to have a really sustainable ingredient and fragrances, since we are using pre-engineered cyanobacteria that is able to secrete sucrose, and therefore these molecules are produced from the CO2 of the atmosphere. So I am now done with this little presentation. Thank you for your attention, and if you have any questions, I'll be glad to answer.
Nice talk.
Thank you very much for your presentation.
Can you just, sorry, can you just open the slide before?
Yeah.
I just take a screenshot and post it on Twitter just to advertise your project.
Okay.
I wasn't fast enough. Thank you.
You're welcome.
Yeah, now you get even free advertisement for your project.
I would ask whether the synthesis pathways for the aldehydes and the terpenes are already working in the organism, so that you only have to manage the cultivation, or do you also have to teach them to make the compounds?
No, we just got the strain of Synechoccus elongatus that was able to secrete sucrose, but for the rest, for the terpene pathway and for the aldehyde pathway, we have to engineer it. Actually, for the terpene part, we are using Saccharomyces cerevisiae strain that is capable of synthesizing lycopene, which is a pigment, red pigment that is in the tomato, and is the precursor of some of the terpenes that we want to produce, but we have to engineer the yeast and the cyanobacteria to produce the molecules.
There's a question in the chat. Does Staphylococcus really excrete enough glucose into the environment?
It's actually a quite tricky point. It has been demonstrated that this kind of co-culture system can be established and can be stable for E. coli, for Bacillus subtilis, and some other species. For the yeast, it can be quite a problem depending on the yeast strain that you're using. Some strains are not really able to grow at very low sucrose concentration, but the strain that we have should be able to do that, and we are also using our dry lab to try to help in the cultivation setup and to see how much cyanobacteria do we have to grow before inoculating with the yeast, and to see all these parameters.
Just a comment, have you been in contact with Danny Duquette?
Sorry?
Have you been in contact with Danny Duquette from Michigan State?
No.
Okay, just write it in the chat, because he actually is like, he created the CSCB strain, and then they have also a co-culture with yeast, but they have like a specific yeast strain. And you might want to just, we're going to contact him, just get an idea. He might be able to also send you the strain.
Okay, for the strain, we actually have it. We obtained it from the Prakrasi lab in Washington. I don't know if he was in this lab or, but yeah.
He was at MIT before in Pamela Silver's group. Yeah.
I think there were two research works. Some were using S. elongatus UTEX, and some were using the PCC 7942, and we are using the UTEX one. So we are hoping that its fast growth would be more beneficial for the co-culture. So, but I can get. Thank you for the.
It's just maybe a good like integrative human practice. Just get a talk with him.
Thank you.
He's usually always involved in Cyanobacteria project at iGEM, like more or less every year.
Okay, thank you very much.
I have one really quick doubt about that, but it's because the terpenoids especially are usually toxic to cyanobacteria because they can just penetrate the membranes and make a mess there. Have you already thought some way to contain this possibility if you are cultivating both organisms together? Because I think at least one team from India, Isarpun, I think it is working on the same approach. Just co-culturing Synechococcus with E. coli, I think, for just changing sucrose for this kind of strategy. So have you thought some way to maybe modulate or simulate this co-culture strategy?
I think that actually the co-culture is quite beneficial for the two organisms. That there have been some papers that show that they work in a kind of symbiosis. The cyanobacteria generates a lot of oxidative species, and the yeast is capable of degrading them. So, and that's also the case with E. coli. And so this kind of co-culture between an autotrophic and heterotrophic can be beneficial, but yeah, it is actually a quite interesting point to.
Thank you.
We have the second. There's also the question, how do you want to capture the volatiles?
Yes, so our final goal would be to use a continuous cultivation system and to be able to actually extract the volatile molecules during the culture. Since these molecules actually are volatile, they escape naturally from the liquid. And there are some processes that exist, especially using evaporation, for example, so membrane processes, and you are actually able to get very pure extracts of these molecules, and it's going to be an important part of the project to see if we can actually get from the fermentation supernatant the molecule in a pure way of or to optimize this kind of process.
Also a comment on this, we also working with terpenes, triterpenes in Ilka's lab, and what we do is we have like a dodecan layer on top of our culture, the cyanobacteria don't like it that much, but they still produce this, and the dodecan basically sucks up all the fragrances and all that stuff. So then you can just like take off the layer of dodecan, and this is also stuff you immediately put on the HPLC and all that stuff. So that might be an idea to just have a layer of this on top. It might be a problem for the co-culture, or it grows slower then, and they don't like it, but then you basically capture all the fragrances.
Yeah, it's also a great possibility, but we actually thought we could use this, but for the sustainability of the system using organic solvents, but it's a possibility if we most likely will do the 2 options and see which one is the best, and how we can, if we can recycle the dodecant, it's actually possible to still have a sustainability.
Yeah, I would just do it in parallel and have it as the first way to actually measure the violet fragrances and all that stuff, just to get the data on it.
Yeah. Okay. Thank you.
Great.
So who's next now? I think Bielefeld needs to present still, right? Or is there any other team?
Bielefeld, go!
Yeah, nobody else wants. So, so, okay, hi everybody, I'm Julia from the team Bielefeld Civitech, and I would like to introduce you the topic of our team 2021, and it is called PLANT, which stands for Plant-Based Ligand Activated Noxious Agent Tracker. And the problem we want to solve is that we have many uninvestigated possible storage sites of chemical weapons from World War I and II, and the map on the right shows you here places in Germany where contaminated sites might be located. But chemical weapons might not only exist at these locations. There are possibly a lot of different places where ammunition was lost. Therefore, it is also very likely that chemical weapons got lost in various unknown locations. Currently, the German federal government makes no major effort to investigate these sites and to remove the chemicals. Due to time, a likely situation is that the containers leak and contaminate the ground, and our system could help to provide long-time surveillance of possible storage sites in order to warn about leaking containers with toxic chemicals. And this is where our project comes in. We want to develop plants that can detect chemical weapon degradation products, and the plants can indicate the presence of a certain chemical via a signal. And the illustration shows how a system should look like in the end. We have a perfectly healthy green plant on the left side, and then we add a chemical weapon degradation product indicated by the chemical weapon warning symbol, which you can see in yellow here. And this leads to a signal that is visible for the naked eye. In our case, the plant is supposed to turn red when it is in contact with a certain chemical. And of course, it would be difficult to teach our plants from scratch how to detect specific chemicals, but luckily there's a publication where the authors created a plant that can detect the chemical TNT. And on the right side, you can see the signaling cascade for the TNT detection. The cascade is a modified cascade from bacterial chemotaxis. And in the modified cascade, TNT binds to a computationally designed TNT receptor. And this complex then binds to a transmembrane protein and activates the histidine kinase. And the histidine kinase phosphorylates a transcription factor, and this transcription factor then translocates into the nucleus and activates the expression of our target genes. Now for our detection system, we want to use this cascade and exchange the receptor to specifically bind our chemicals of interest. And we also want to exchange the reporter. And I would also like to give you a short overview of the different parts of our project. First, we have a computational protein design part where we design a receptor that our chemicals of interest specifically bind to. And then we use different systems to test the designed receptor so that in the end we can establish the whole signaling cascade in a plant. And we also establish a reporter that is triggered by a modified signaling cascade to get a visible output so that our plants will turn red. To summarize, we want to create a plant that indicates a contact with a chemical weapon degradation product by changing its color. Yeah, thanks for listening.
Okay, thank you very much for the presentation.
Is there any question? If not, can continue with the next team, which is ASU.
Hello, we are the ASU iGEM team. Although we don't have a team name really decided yet, we are looking at arsenic reduction using Chlamydomonas reinhardtii. So arsenic contamination in groundwater is a problem worldwide. In particular, in Arizona, there's lots of overmining and improper disposal in certain rural regions, which can be essentially dangerous and lead to arsenic poisoning for certain tribal regions or rural communities. So we are hoping to sort of tackle this problem using biological systems and utilizing some native arsenic uptake mechanisms in climbing. So like I specified, Chlamydomonas reinhardtii is capable of sort of taking in or sequestering arsenate from the environment, reducing it to a less toxic form of arsenite, and either sequestering that or releasing it back into the environment, improving its survival in those high concentration environments. And while this system has been shown to work pretty well in different concentrations in Chlamydomonas, the application of that in E. coli has more or less just been seen as looking at just the survival of E. coli in these arsenic contaminated areas. So what we're now hoping to sort of do is improve the capacity of Chlamydomonas to increase that bioremediation process and hopefully reduce or remove more arsenic in our water. So we plan on looking at specifically 3 genes, which would encode for arsenate reductases, metallothionein, and ferritin, which can essentially remove or reduce the toxicity of our arsenic in the water, as well as improve our sequestration. So it's actually able to bind most heavy metals, these metallothioneins and these ferritins. And we'll hopefully be able to express these different proteins within our chloroplast genome, which can help us improve our expression and potentially lead to some interesting or more effective methods of bioremediation for this. So we are still currently working on developing some protocols for the analysis and quantification of our arsenic reduction. We're looking at potentially using some liquid chromatography or mass spectrometry, just so we can actually quantify how much arsenite we've isolated in our biomass, as well as what's left in our concentrated water. Potentially, there are some reductase assays or some colorimetric assays as well. But like I said, this is still sort of a little further down the line. In the end, we're kind of hoping to implement this system into contaminated areas, rural regions, as well as industrial wastes. So we're actually able to sort of generate these maybe filter membranes, which can allow water to pass through our membranes and potentially allowing any sort of heavy metals or arsenic to be absorbed within the cells and isolated, so we can then dispose of that contaminated biomass more safely.
Okay, are you done or? Yeah. So thank you. Is there any question for that talk? If not, I would have a question. Okay, then I will start. What, even if you don't do it, but what would your final vision be? How to apply the clammy? Let's say you have a clammy strain able to take up this arsenic. Would you like envision to put it in the environment or like in the water directly, or like or would you have like a huge bioreactor which can like take up all this contamination, or what's your final vision how to get this out of the water?
Right, so we're currently sort of developing what we hope to at least generate a larger sort of reactor or just a pump module that could process large amounts of volume, because we're also sort of in the process of determining exactly what concentration our clammy would work best in. And I suppose with enough testing, we could end up determining how much arsenic are we able to process in one go or one batch, and then potentially making this more streamlined process for, like I said, either rural environments or industrial waste.
Okay, thank you. Is there any other question? If not, is there actually any other team which hasn't presented and wanted to present or has prepared anything we missed, or if so, speak up or write in the chat.
Also, if there is a team that hasn't prepared anything but just wants to say 5 words about their project, also feel free, just
yeah, of course,
spontaneously. But yeah.
Oh,
I could maybe give, talk 2 sentences about our project.
Cool.
It's a national holiday here in Finland, so I don't have a presentation. I've been, yeah, it's a public holiday. Anyway, so we're Team Abo from Turku in Finland, and we are trying to, with our project, aims at aiding the situation of the Baltic Sea through developing a microbial wastewater treatment system for the degradation of pharmaceuticals that end up in the water and cause major environmental issues. So we got some inspiration from last year, Kaiserslautern, then there was Stuttgart and Darmstadt. So we are continuing with the diclofenac degradation, and we're then comparing 3 different lac cases, so enzymes that degrade diclofenac in different conditions, and seeing which one would be the most potential. The previous teams had many issues, so we are now we have a wild card anyway in our project then to see how that would work in pH 7, which is for the wastewater. Yeah, so that's, and then of course, yeah, why we are here. So we are doing it in E. coli and in Synecocystis sp. PCC 6803. So we're trying to get it transformed into both and see how it works. Yeah, that was my super quick introduction to our project. Hopefully some more details later on in the season.
Nice.
Awesome.
I also really like that like teams are picking up old problems which haven't been solved completely. So it's always nice. You don't have to reinvent the wheel with a project, but you can also continue previous efforts. I like that.
Yeah, and of course, we first came up with the diclofenac, and then we checked, has it been done previously in the iGEM? And then we realized that, oh, wait a minute, there's been 3 teams last year doing it. So it wasn't directly, we were looking at their projects and then it came up with ours. But
yeah.
Came up with ours and then we saw, and then we are not trying to copy them. So then of course we have different chassis and we have different enzymes and a bit different experiments to not, of course, we cannot, we don't want to repeat anyone's work, of course not.
Yeah.
Yeah, but still cool to continue from where they ended up, and they hadn't really compared that many, so, or the specific ones that we have. So that's will be interesting to see if we get some more promising results than what they got previously.
Cool. There's one more hand raising.
Yeah, thank you for the opportunity to present the completely unprepared team presentation. Yeah, so we are the LMSU team from Moscow, Russia, and unlike the other teams, we are not solving any particular problem in the be it local or global, and we are introducing the Arthrospira platensis strain as a novel chassis for the synthetic biology, as we have discussed in the Cyanobacteria room. Yet it is the only generally recognized as safe strain of cyanobacteria that is possible to eat and to sell as food or feed. And in our project, we are going to describe our chosen host via genomical, physiological, and biochemical analysis. We will perform, hopefully, we'll perform the genome assembly and annihilation, as there is no like Arthrospira genome at NCBI at all. We will try to grow it on different media, and this ratio of the storage compounds as lipid, starch, and proteins will be measured as a response to different growth parameters. Then in the genetic part of our project, we will going to try to propose a transformation methods, and we will and I have collected some of ideas also in the bacteria room for some tweaks or completely new approaches. We will collect some data on the transformation rates, and if we succeed, we will try to analyze the behavior of readily available biobricks parts. And also we will try to analyze the foreign DNA and the heterologous protein degradation rate. And so in order for the researchers to know how long the plasmids or linearized DNA live in the cells, and for example, if the Cas9 protein will be transcribed and translated, and in order to like act on the genome itself. And then after that, or I should say in parallel, we'll present the proof of concept solution, which will include the implementation of our transformation protocol. We have designed the custom optogenetic regulation system, and under that, we will propose the bioproduction solution for space, and we will contact Melissa and from European Space Agency and NASA in America. We will enhance Arthrospira with some flavor-producing genes, for example, vanillin or some other, or for example, maybe the lemon flavor or menthol flavor or something else. And yeah, that's a lot of work, and I hope that we will collaborate with some of you. We are collaborating with the Madrid team for C-Fuels in the part of analyzing the promoters, and we have proposed a guideline for analyzing the relative strength of promoters in living cells using fluorescence, and we will be glad if it is used by more teams than just 2 of us. Yeah, thank you.
Nice. I really like that there's still teams like working on like basic chassis development. I think this is really important, especially as you mentioned that spirulina is not very well established, and also the transformation protocols are really, really tough. So glad that you are working on it.
Yeah, definitely. I mean, given that it is already kind of in the market, if we would live in a world where people want to eat GM food, it would be super cool to like engineer them to have like different flavors and everything. It would be kind of
Yeah,
nice.
and apart from flavors, it possibly can be enhanced with just about everything. For example, the zinc or magnesium uptake pumps will enhance it with micronutrients, and yeah, also it could produce, as some of speakers has told, it could produce edible medication, and some of our some of papers that we have read produces the whole like camel antibodies, I suppose, which have some therapeutic action. Yeah, so it's really promising, and we'll try our best. Thank you.